You are on and you can go on, Enrico. Thank you.
Okay, I don't know. I'm on in my screen.
Yeah, absolutely. And you can share your screen.
Okay, I'm sharing now or not?
You are perfect. Yes, go ahead.
Okay, so today we talk a little bit about correlation and correlation along a line, and Susanna did a very nice presentation about that. So I will talk about something which is called image mean square displacement, and you will see that. So the mean square displacement generally is a method used in single particle tracking to figure out the kind of motion of a particle. So let us see what is the basic idea in using the spatio-temporal correlation, so where space and times are together. So in the spatio-temporal correlation, we assume, is the basis of the equations, that given a probability to find the particle as a function of time, that probability will change as a function of time by two factors. One, which will increase the variance of the width of the Gaussian and will increase linearly with time. And the other is that the amplitude has to decrease too in order to maintain the integral to be a probability. And that will go with a more complicated rule, which is here. So the question is, if we can measure all that distribution and the distribution as a function of time, we will have much more information, and that is done by the iMSD technique. So let us summarize most of the technique I have discussed so far, and let us see if we can make a common point of all of it. So when we do single point correlation, we maintain the point, which is indicated by this blue circle, at a given time. And then if we consider we have a certain number of molecules, that time, as a function of time, the molecules will leave that volume, and then we will have, if we correlate that time with that time, or that time with that time, we will get here and here exactly the same, because the same molecules are in the same volume. But if we have from here to here, some molecules are left, so the correlation function will decrease. And then the farther we will go to the average value of molecules in a given volume, which is what we subtract, the famous one that we subtract in the equation. Okay, so this is what happens in the normal single point FCS, which is what I explained this morning. Now in scanning FCS or RICS, what we do is that instead of maintaining the point in the same location, we move the point. You see, I am moving the point. And so by moving the point, two things can happen. Either we move the point very, very slowly, and then it's about the same point FCS, or we move the point fast, like Susanna was doing and so on. And then what happens is that we can have individual volumes, and then by moving the point, you can still study the distribution. So the distribution here, well, it starts from the same number of molecules, so it's supposed to be the same, but we're scanning the distribution. And in the spatial correlation, for example, at the single distance, so we have two volumes, so it's like the per correlation, we can see some of the molecules which are in one volume to go into the next. So those are the basic ideas shown in a graphical way of the methods we use. Okay, so I told you that if we measure GFP in solution or GFP in the cell, the correlation function stay the same, but it moves a lot, about a factor of 5. And people say that this is due to change in viscosity inside the cell. And I say this is not very convincing, because if you measure rotational rate of a molecule of the size of GFP, you will find that actually they rotate exactly in the same way. So let us try to join the different kind of things. So we have essentially a Gaussian distribution, and this Gaussian distribution becomes flatter and flatter as we go with time. And if we have immobile part, so at some point it will change, but will not become zero, because we have some parts which are immobile. And that was discussed in the case of it. Okay, now let us see what is the difference between the iMSD, which stands for image mean square displacement, from FCS and RICS. So let us consider in this column essentially a different process. So the process of a molecule which was originally in one side, it will move randomly and will explore a different side. So here is the molecule, but at this time, the molecule cannot go to the next side because it's confined, and we say that this molecule is confined. The other, which would be a transient confinement, which happens very frequently, is that the molecule is confined for some time and then jump over the barrier and can go farther. Okay, so let us see what the iMSD will measure, and I will show you the equation for that. So as a function of time, if we have pure diffusion, we will measure exactly what this expression was saying. So as a function of time, the Gaussian expression will increase linearly with time. Okay, so by changing linearly with time, we can see that the slope of the curve will depend on the diffusion coefficient. So the faster is the diffusion coefficient, per equal amount of time will go farther. And this is measured, so the iMSD is measured in micrometer square. And that is time, so micrometer square per second is a diffusion. Okay, and that works is, well, there are many experiments that show that works, and this is the simulations versus the theoretical values, and they are one-to-one. If you have confinement, what happens is the following, that the particle will start to move exactly in the same, but then cannot go as far as as the diffusion will drive them, but they are confined. So, for example, if they are confined to stay in 400 nanometer, we will get that the maximum distance will be 400 nanometer. And then if it's 300, it will be 200. If it's 100, it will be 100. So we can measure the size of the confinement region just by measuring where the particle will go. And here is, again, a theoretical means what input to the simulation. This is what the simulation recover, and is absolutely real. Then we have a different kind of motion, which is a transient motion. So a transient means that for sometimes it will get confined, but then as the time goes on, it will go out of the confinement region, and then we will have like a free diffusion. And we can calculate that probability just by measuring the initial part and the final part. And so measuring all those quantities, which is available in a measurement of FCS, we can obtain all information about not only the diffusion, but also the confinement and transient confinement. So let us see how that appears in the, so the general equation for the iMSD function is an equation which depends on the coordinate x and x, what this is in Greek, x and y, and the time. So this is the expression that we do. And then we have seen that this expression can be divided in different parts. So there is a G0 term, there is a excitation volume, so the volume of excitation, and then the probability to find the particle in the given position. According to Fick's law, this probability should be this expression, well, it depends on the dimension, but it will be in this expression. So in the MSD, we divide the various parts. So we have that the correlation function will be a function of the temporal correlation. So if we stay at the same time, it will be just the diffusion. Then we'll broaden the probability by whatever is the Fick's law, and then we'll have a constant offset. Actually, this is very interesting, and I will discuss what that means. Okay, so let us see the effect of the size of the particle. So there is an effect of the size of the particle, which means that when we measure the intensity of that particle, even if the particle can move at time t equal to 0, so the intercept of time t equal to 0, it will not be exactly 0 because it will be at least the size of the particle. So if we can measure that value, which is contained in this equation, so we can measure that value, then we should be able to measure the size of the particle. How precise is the measurement? Well, it's a lot precise. So if we do simulation again, if we have particles which are, this is 100 nanometer, this will be 10 nanometer, this is the recovered value, well, it's more or less, follow in a line. So that means that between 1 micron and 1,000 nanometer and 10 nanometer, more or less, we can recover the size of the particle. And this is very useful because that can give also information about size of clusters eventually. So I would say the particle size down to about 10 nanometer in this technique can be obtained with very high precision. Note that the sampling is low. A small particle will appear big. So if we expose a camera for some time and the particle is moving, the particle will appear big. So we need to sample as fast as we can in order to obtain this information. Okay, so let's go now to application of that. And the application of that is, there is a very interesting application, which is by the work of Kusumi, which is, he makes the hypothesis that in the membrane molecules, proteins will move, essentially just membrane proteins will move depending on the cytoskeleton, and essentially there is a cytoskeleton, so the particle will move in a given part and will be transiently confined, and then eventually will jump over and then will go and more. So if you look at the data, so we have this data. So if we have essentially in the absence of electron cooling, which is a molecule that will destroy the cytoskeleton, we will have this behavior. So which means it's a transiently confined, as we have seen before. But if we have add electron cooling, then becomes almost free motion, and then we have free motion. So that was very nice, and you can obtain essentially the same result that we obtained years before by Kusumi. Okay, and then here we start as a function of temperature to show that this activation of a barrier, they didn't have this information, but we show that you can obtain an Arrhenius plot and calculate the probability to go over the barrier. Okay, now let's go to the important part I want to discuss here, which is this idea that you can do line scan faster and faster. And let us see what's happening. Okay, so suppose you have a particle which is here, and then you scan over the particle. If the particle is not moving, you will see exactly a Gaussian, which will be the point spread function. So it's really how the point spread function is passing over the particle, which is mobile. But according to the RICS and everything that we see, if the particle is moving, you will have a narrow size of the correlation function. And then if the particle is moving, so the speed of scanning is much larger or smaller with respect to the movement of the particle, you will end up with something which is very, very narrow. So the line that we will measure will be will give us a direct information about the motion of the particle, about the diffusion of the particle. So we can do this experiment now because it's scanning. So we can sample at about a microsecond, and then we can go much lower, and then we can measure as a function of the speed of scanning, for example, the diffusion of GFB in water, which is about 90 micrometers square per second. And you obtain this line. So don't forget this line, which will be in green in all the data that I will show. So the iMSD along a line shows that actually the GFP in water is a diffusion. In fact, is linear. And the slope is about what is supposed to be for iMSD. Okay, for the GFP. Okay, now let us see if we can do the same thing, but measuring in cells. So we can choose a part which is in the cytoplasm of the cell, or a part which is in the nucleus of the cell. So that line green, I copy from the slope from the previous graph. So it's just what is supposed to be if we have a GFB in water. And this is in a cell now. And so notice that it deviates, but then it becomes like water. And becomes like water only if we can sample very fast. Otherwise, we will not see that value. And that is very rewarding because that shows that, yes, in a given a small size, and here is the size of the movement, so 10 to the minus 2 micrometer square or 10 to the minus 3, it moves like in water. Has exactly the same slope that the molecule is in water. So if you are a very small volume and which is the same thing, or very fast time, you will see that GFP moves like in water, which is what we always knew. Okay, now let us see what happens. Sorry, let us see what happens if we have. So remember, the green curve is just GFP in water, and now we are in the nucleus. Well, in the nucleus, we never get to be equal to water. Might be those points, but within there. But you see, it's always different, and so which means that it never reach in the nucleus. So this is the region we're exploring, the motion of like in water. Okay, so what is that, and where is that coming from? Well, we say, well, might be in the cytoplasm we have structures, so we have cavities, and so let us use a molecule like GFP. So remember, the green line is just GFP in water. So let us use GFP in this BSI solution, which is that point, and you can see that never changed the slope. So it's always the same. But, and then, so this is a solution, the fit, and this is in different amount of GFP. So the slope is after diffusion coefficient. So never change. But if we have a polymer, not BSA, but the polymer which has cavity which have water, and then polymer and water, then we can see this behavior in which we say that some region is start to have the same slope than in water. And that size of the cavity corresponds to the physical size of the cavity we can measure with other means. So that means that the interior of the cell, at least the cytoplasm, is made of cavities where the GFP molecule can move very rapidly, like in water, which is the result we obtain from the rotational rate. And to give you an idea of how good is the system, so for example, this is Alexa 488, which has a diffusion coefficient of 428 in water. And yes, it moves exactly with the diffusion coefficient. You can measure very accurately those values. And this is for beads, which simply means that they are also diffusing. Hello? Yeah, which also means they are diffusing, and they are diffusing more or less with the same speed. So there was a question I had about FRAP, what is the difference between FRAP and the other techniques. So let us make this graphic, that's the last one of this presentation, in which I showed the region in which you can apply FRAP. So at the best, you can go to 10 to the minus 10 to the minus 4 second, or I will say that it's more limited to that region. But anyway, but it's diffraction limited. So you can make a spot which is smaller than the diffraction limit. FCS is also diffraction limited, but the iMSD and RICS is not. And you can go to much faster time, which also means much smaller distances. So look at movement in very small distance. So that shows another difference between the FRAP technique and what you can measure with those spatio-temporal techniques, RICS or iMSD or PCF and so on. So I will stop here for this presentation. I wanted to show this data because I think, you know, what complement very well the discussion that we had when I give my lecture, I say, where is that coming from? So there is nothing magic. Simply, it's the structure of the cytoplasm, which is different than the structure of the nucleoplasm, which is different, which is more similar to a polymer with cavities. Okay, so I will stop here and answer your question and then go to the next application.
Thanks, Enrico. Very nice. I'm trying to see if there is any question in the chat, or if anybody want to.
Or maybe I have other two things. I think this is a comparison with a technique that has steady FCS, in which we can go slower, we can go smaller and smaller volume, and this is by iMSD, so more or less the points are very similar one to the other. So this is just, I have a question if the public doesn't have any question.
I have a question. If you think, you say, I mean, you need to go really, really fast in order to catch basically free molecule diffusing in
a small distance.
Well, this was my point. I mean, if you have a camera that you cannot run at that speed, how you can handle in order to get the very fast, like I don't know, fluorescing in solution.
Okay, so there is a different kind of technique which is limited, and this is the so-called K-space technique of Paul Weissman. Okay, so instead of measuring the correlation in the real space, like we are working, so our coordinate X and Y of the real space, you take the data, make a Fourier transform, and then you look at the various components of the Fourier transform statement. Okay, what happens is the following. That suppose you have a camera which is 512 pixels. So the minimum area that you can measure is 1 pixel.
Yep.
Okay. The maximum area is the size is 512 pixels.
It's the chip.
Well, it's the chip, it's the size of the chip. So you are very much limited because you can go a factor of 500 there.
Yeah.
And even if the camera is 1000, generally you can go slower than the camera he was showing, but it's limited. Here, you know, if you see the number of decays you have, so it's 1, 2, 3, 4. So essentially in the other region, there is no limit. Yep. But you can never cross this line. So you stop at that point. And even if you use the space, you exchange the space for frequency using the spatial Fourier transform, still, you know, you cannot explore that enormous change that you have here with the line.
Yeah. Yeah. What I was thinking was, I remember when we wrote the paper for the comparison between path correlation and iMSD, and you asked me to make a plot, which was, well, either you can, if you have something that is moving fast, either you have to scan fast or record as fast as you can, or you have to go as farther as you need in order to catch the diffusion. So the speed.
And this is what I said, with the camera which has 500 pixels, you cannot catch it. You never reach that. Yeah. It's the same question, just in a different way.
And the other thing is, yes, speaking about Paul Weissman approximation, maybe you can just stand a little bit different in between iMSD and the spatial correlation that he does, because basically instead of looking at the spreading of the point spread function, he looked at the shift of the point spread function in order to see the directionality of the flow, right?
Well, PCF of course gives you the directionality. Okay, but it's based on scanning, and the scanning is much faster. So the only technique which is similar is using the ARRI camera. And you say that this is like, I will call that a nano camera, which is okay. In fact, in that case, the frame rate is 1,000,000 frames per second.
Yeah, yeah.
So yes, at 1,000,000 frames per second, you are here. And you can measure that distance, and you can measure that very well. But you have to go through a different kind of camera and use the camera in a different way.
Yeah. Okay, great. I don't see any. Oh, Ernesto has a question.
So I have written 4 questions here. So we only discussed the first. Okay.
So now, Ernesto.
Hey, Enrico. I just wanted to ask you whether you can more or less have an idea of these confinement spaces where you have in the cytosol for GFP, if you can have an idea of the size.
Yes, yes.
Let's say, so you have to scan this length at this speed to get the diffusion of water?
I mentioned that. So when it becomes like water, this is your size of confinement. And your size is, well, this is square of the size, but will be 10 to the minus, well, it's about 50 nanometer, you see. So you have to take the square root of that number, okay?
Yeah, and well, having that, is it homogeneous all around the cell? All this?
No, not really. So
I mean, not saying within the nucleus, but within the cytosol.
No, it's not. It's not, because if you get the mitochondria, or they're much, if you have to go far. If you get a yard or the other things, it's changing. But you know,
yeah.
you're measuring by the intersection of those 2 points. This is water, and this is interception. So that point of intercept is when the 2 things start to go together. And that happens in different positions. You are absolutely right.
Well, that's great, because you can say, okay, within these places you are more confined or less confinement. That's great, and with a determined pore size, let's say.
You get the average size, of course.
Exactly.
The size is the average.
Yeah, you also consider, Ernesto, that you are an averaging a region, because this box, it has at least 32 or 64 pixel square.
Yeah, but the per correlation function, you don't have this limitation in some way, because you move the point.
Exactly.
Right.
So here also you have a line scan, but the line scan is extremely powerful, because you can go, you know, count order of magnitude, and in this side there is no limit. And I try to summarize in this graph, so that the normal technique and the FCS are here. But then when you use this line scan, you open up the space. Completely.
Totally.
Wednesday, Lorenzo will talk about this correlation within the nano camera.
With the nano camera, which is essentially the same idea, but instead of being a line, you have a surface.
Can I ask the last question? Did you try this line scan in the plasma membrane with dyes?
Yeah. What do you mean? I show, well, maybe. So this is in the plasma, cytoplasm.
At the plasma membrane.
In the nucleus, never goes. So.
No, at the plasma membrane. You know, if you have
no, we didn't
dyes to see, you know, let's say, I don't know, probably you can label with Gen1 with fluorescent cholera toxin.
Yes, I know that. No, okay. We haven't finished the work. There's still a lot of things to be done.
Yeah, exactly. Well, I'm coming up with the experiments that I would love to do. Okay.
Yeah, no, so of course, you know, the famous structure in the membrane are supposed to be very, very small and very, very fast. And we are, well, this is a limit in some way. We can go maybe another factor of 10.
Okay.
But we cannot go much lower simply because you don't get very many photons. But you can go, in terms of size, another factor of 10. And that's in speed, a factor of 10.
Okay, cool. Thanks a lot.
Okay. And we're doing that now. Lorenzo will give a talk in which we get not only, so this is just intensity, but we get lifetime and spectral information. So that will give much more information. But you're right, you know, will be an ideal things to study membrane. And iMSD is very easy to do because you can do in the TIRF instrument.
Okay, Enrico, let's move on to the last talk.
Okay, let me see if I can. So last talk was my presentation, application number 3.
Yeah.
Okay, that was the one it was supposed to be.
You have to replace the screen that you are sharing.
Stop sharing and reshare.
Yep.
Okay. That one? Is that okay now?
Yes, perfect.
Well, I have to make it presenter. I have to go out of the presenter.
Yeah.
Okay. So now it's supposed to be better. Okay. So in this application, I want to talk about something which we published some years ago with Luca Lanzano and Francesco Gandarelli, and we published as a letter in Biophysical Journal because we were in a hurry. And I don't think has really a lot of public, but I want to mention something which is very important about correlation functions. Okay, so we already discussed all this stuff, and then I have the scanning FCS principle, so which was discussed by Susanna, and I will say a few more things about that. So Susanna mentioned that I will talk, but I didn't at that time, about scanning rapidly a line or a circle, which is faster, and then come back to the various points. And then she talked about dividing the data in the carpet and plotting the carpet and so on. And the other thing was to obtain, so how that is obtained, and then to obtain information about the diffusion of motion. Okay, and so the question is now, okay, what kind of correlation function can we obtain from this kind of analysis? And so let's go to the point of the separation and the other point, and let's go to the problem we want to find. So she talked about being overlap or no overlap in the points, and both are important if you want to see very fast process. Okay, and this is, for example, when you have particles, and so that's part of our software, obtain all this stuff. But now let us discuss what happens for example, when we do the intensity at one particular column of this function. And on one function of this thing, we obtain what is just a normal correlation function that we will obtain if we have essentially a particle diffusing. And that is what she wanted to show. But actually, there is much more information here. And now let us open a different kind of application and see the information that we can obtain here. So somebody asked about if we are doing GUVs or other things. The GUVs move, and well, I don't want to discuss all the part. Okay, so let us start again with the nuclear pore complex. And now in the nuclear pore complex, we have some ternary complex made of importing alpha and importing beta that will transfer in the other side. And the question is how the transfers occur. I show that NLS termination in the molecule makes a difference. But let us see what is the difference and how big is that difference. Okay, so first of all, how are made the nuclear pore complex? Well, the nuclear pore complex, we know most of the proteins. So there are 30 different polypeptides, so proteins, and each in multiple copies. And actually, it's an octameric structure. So there are 8 subunits of those many complexes which are acting all together. And they transport through a nuclear pore complex. You can transport ions, RNAs, of course, very important, proteins, entire ribosomes, etc. And to give you an idea, this is more or less in scale. So this part of the nuclear pore complex is about 50 nanometer. And this side is about, the total size is about 150, 200 nanometer. All those are important things. And there are also important things to know that we know the proteins, we know the location, we know the octameric structure, and so on. So let us see what kind of things, well, we already discussed what kind of things can go through from the nucleus to the cytoplasm and so on. But let us see what we know about it. I jump to 10. So jump to 10 again, involuntarily. Okay. Okay, so I want to understand how the transport through the nuclear pore complex occurs. So the passive transport, which is simply due to diffusion, is bidirectional, so occurs in both directions, independent, is regulated by molecular size. As I said this morning, what was morning for me, about 60 kilodalton molecule can go through the nuclear pore complex, but it's energy independent, but it requires a lot of time, simply because the length of the channel is long. It's about 200 nanometers to 300 nanometers, and this is the time that is measured. So by diffusion, it takes about 150 milliseconds. And this is more or less the theoretical value. So active import is instead unidirectional, so can be from the nuclear, from the cytoplasm to the nucleus or reverse. Is not affected very much by molecular size, so we don't know how, but it's energy dependent, so it requires GFP. It requires ATP, sorry. Okay, so let us see how that occurs. So in the cytosol, there is a low concentration of NGDP. In the nucleus, there is a much larger concentration of NGDP. And this is the thermodynamic free energy that allows transport through the nuclear pore complex. In the cytoplasm, in order to get into this nucleus, you need to form at least a tertiary complex, actually much more complicated than that. And then there is some sort of interaction of that complex with proteins inside the nuclear pore complex, and then magically the system is, the molecule of the complex is found in the other side. I say magically because the mechanism that people have suggested is that there is known FG proteins, or reaching glycine and phenylalanine, that are in the interior part here of the nuclear pore complex. And the basic idea is that that net can be broken, and so the hole opens up, and then molecules can go through, but still by diffusion. Okay, so if we measure the time it will take to pass through, we can exclude model. So there are three kinds of models. So one is essentially the destruction of the hole. The second is the phase separation. And the third is the saturated phase model. And let us see how that will happen. So, so clearly, we need to know each individual molecule, how it goes through the nuclear pore complex. And this is the most important thing. So I already showed a picture that shows that essentially you don't get any information about how you go from one point to another, other than the time it stays in the nuclear pore complex. Okay, so I discussed about the pair correlation function, so I will not repeat those things because I already discussed, and go directly to the problem. Okay, so, okay, is the question of how selective getting, which is one of the model for opening up the nuclear pore complex, occurs. And so we know that if the system here is highly disorganized, those proteins don't form any structure, and how it happens that it's open up. And it looks like, if you have to draw in 3D, it looks like really a spaghetti without sauce or something like that. Okay, so this is, so what is the basis of this selectivity? How can distinguish that NLS is necessary to go on one side or not? And so the question is that there is an energy, a free energy, part is binding and part is entropic, and a molecule can reorganize the structure. But after reorganizing the structure, no matter what is done, it has to diffuse between one part and the other. And this is the point. So, so if you get the proteins which are inside, and then you can form a gel of those proteins, and then you can see that that gel at some given temperature, and depending on it can interact with some molecule and become like a fluid. And then they say, well, that has to be a mechanism, because that becomes a fluid, and so will allow molecules to pass through. And this is shown here. For example, you have a buffer side and the gel side, and then you add a protein which is marked with the DS red, and then you can see that after a very long time, everything you have in the buffer side will be found inside, and so on. And then based on that, they say that is the interaction with this protein that will break the bonds, and then eventually will form a liquid, and things will pass through. Another kind of model, which was proposed by Lim et al., says the following thing. So you have in the you have the GFP, the FG proteins, and then you approach with the AFM tip, which has this protein interact with the GFP, and you can see that as you get closer and closer, the extension of those protein will change, and then essentially you form something which is from this side to the other side, and then you say, well, this is the process that occurs. But remember that the only thing you have done is simply decrease the size of those molecules. Still, in order to go from one part to the other, you need diffusion. And there are pictures that shows that and so on. And so the question is diffusion versus direct motion proven or not? And the question is absolutely not, because all the diffusion, it depends on, if you don't measure the time, you will never been able to figure it out if it's diffusion or it's active mode. Okay, so let us see if we can start with a new technology. So here, so in green is importing beta. Sometimes it's called a caryophyrin because it's related to the motion. And this is the image that we obtained with my microscope that shows that actually some regions are enriched in importin beta, and then this is where a nuclear pore complex is, at least at this resolution. Okay, so the question is, can we focus on one of those nuclear pore complexes and see the molecules passing through? So, and how they pass through. So first of all, a nuclear pore complex in about 5 seconds moves very, very much. So you cannot do an image and figure it out which is which, because that shows that images at 0 seconds, at 5 seconds, and 9 seconds. So it's completely, has moved completely out. So what we need to do is to track the nuclear pore complex. And so the fluctuation analysis will not give us very much because the object move too much. Okay, so in order to do that, we will use the orbital tracking. I will not describe the orbital tracking, but the orbital tracking, you orbit a particle, in which case will be a nuclear pore, an entire nuclear pore complex. And as you orbit the particle, you will center the particle exactly at the center, in which case the intensity will be a constant. So the modulation, what we call this amplitude of the modulation, will go to 0. And this is the method we use. Okay, so let us see what happens. So if the particle is exactly at the center and we orbit around, the intensity will be constant. And so if it's not, the intensity will start to increase as the particle close to the orbit, and then we continue to increase up to about a certain factor. And then by measuring the modulation, we can feedback the system in order to bring the modulation equal to 0, which means we are centering exactly the particle. So this is in a few words how the method works. Okay, so now let us see. So we see the nuclear pore complex. So we make an orbit around the nuclear pore complex, and then we are look at this orbit. So in one side will be, for example, the cytoplasm, and the other part will be the nucleus. So any molecule that will pass through will appear in this part of the orbit first, and then will appear in the other part of the orbit first. So that was the basic idea. Okay, so let us see if we can go through. So now I draw essentially the same thing, but in a more realistic scale. So this is the orbit that we will do, and this is the nuclear pore complex in the way we see. So we are focusing on one, just one. this will more or less be the size of the point spread function. That will be the size of the nuclear pore complex. And we do the orbit with the time, which is 0.5 millisecond per hour. So every 0.5 millisecond, we do the complete orbit. So if the orbit has, we measure along the orbit, for example, 128 points, every point will be about a microsecond or something like that. So this is how the experiment will be done. Okay. So this indicates that we track, so the X and the Y move, but the modulation stay constant, and simply say that we can track. Okay, now here is the point. So if we do a autocorrelation, just a normal correlation function, and we have the karyophylline alpha and beta, which are marked, we never see a correlation function which is starting high and going low after a given amount of time. We see dots in the ACF carpet. And where are those dots? Actually, if we count, maybe we get 8, 1, 2, 3, 4, 5, I don't know, a certain number. And then if we analyze one of them, we see something very, very absolutely peculiar, that our autocorrelation function is made of a peak. We say a hump. And how an autocorrelation function can have a hump? And well, I said if it's diffusion, cannot have a hump, but if it's not diffusion, could have. And so let us see in the model what will be the idea. So the idea will be the following. So that sometimes, like this time and the blue, correlate always because always are at the same distance in time. But if other processes occur which are not at the same time, they will never correlate or will not give a hump. So, the idea is that a molecule is there, disappear and return there, and then something can happen in the middle, appears and disappear, appears and disappear, and this is the process that we're measuring. And since although it's random, it's not periodic, has a period, not in the total period, which does not exist, but has a period between the one position and the return at the same position. And this is very, very important. Now, that 2 points can be one side and the other side of the orbit. We have to figure it out now where they are. So we focus only on those processes which have an intensity and then an intensity at the given time. Remember that we are doing orbits. So how that can happen? Well, that can happen if a molecule, for example, while we are doing an orbit is here, and after some time, it's in the other side. And that time is fixed. Okay, and then even if the event occurs random, still will correlate and will give us a correlation which is, given by the time it takes a molecule to go from one side to the other. And if we continue, eventually things will come back. And then we can measure the time. So we can measure all those time independently. And we can make, we can do the correlation in one direction, so point here and the point there, or a point there and the point here. So we can see the direction of the motion and the time it takes for that, process to occur. Okay, so let us talk about the experiment, how we will do that. So we have a cargo that is supposed to pass through, has a GFB, which is green, and then the nuclear pore complex is in red or another color. So suppose we have the two molecules are independent. If they are independent, we will never see dots in the carpet because they are totally independent process, and then we will see only maybe some points with some correlation. So that means that there is more chances to see one passage to a given point of the nuclear pore complex and so on. But now, now suppose that we observe the passing of the cargo by making a cross correlation between the red part and the green part. If we make that, we will know if the cargo which is in green, was here, and then reappears in the other side, because it will change color, and then we can measure how long it takes to happen that. And so I say that is contrary to all the process. So we will have the whole process of diffusion, and then this PCF approach, so the correlation of the point along the orbit, will give us the direction and will give us the time it takes for something which is in one side to appear in the other side. Okay. So, so now let us study this process. So we have the cargo, which is in green, and we have a protein which is just in red, and is pure diffusion. So the green one will have a correlation, this hump in the curve, and that correlation time is 3 milliseconds. And the other one is much, much longer, which means, yes, it passed through, but at a total different time scale. Okay. So now let's go on with that part. And the question is, among the many proteins that we have, is there a protein in the nuclear pore complex which also move together with the cargo, which means some causality in the motion of the cargo? And so we choose a molecule which is NUP153, which is a very large protein, has 1475 amino acids, is anchored near the N-terminal to the nuclear basket of the nuclear pore complex. So the basket is this part, so it's anchored here, and contain a large number of repeat FG, and contain 4 RAN BP2 zinc fibers, and have both the N-terminal and C-terminal that we can mark with the EGFB. Okay, so we have, okay, so it goes back all. So we have a molecule that is anchored in the basket here, and eventually, if it's part of the transport, will go up to the other side and eventually contract again and bring the cargo in the other side. And that will be, we mark in green, and we can mark, say, the N and C terminal. And if we see the ACF, the correlation function of the NUP153 also has a bump. And it takes about 7.5 milliseconds to see so the characteristic bump is 7.5 milliseconds. So which means that every 7.5 milliseconds it goes up and then goes down. And so this is what is happening in this protein. So now we can do finally a cross correlation between the motion of the NUP153 GFB, so in green, and the cargo, which will be in any other color. And then we can see essentially when the protein is collapsed or when the protein is extended. Remember that the extension of the molecule is such the size that will go in the other side. So, so the question is now, so we do the cross correlation, and then we observe what happens about the cargo, and what happens if the motion is correlated with the motion of the NUP153. And essentially, we'll grab the cargo, eventually, some of the cargos, not all, of the cargo, and we'll bring back in the other side. And then we'll release, because here the concentration of the RAN GDP is very high. And so, and then we'll eventually return, and so on. So now we can see in 2 colors what is happening in great detail about this stuff. And so we can measure the NUP53 when it's collapsed, and so in the carpet, so every time we pass here, and once in a while it extends and then returns to the position. So this is the position in the orbit, we see that. So we see extending in 3.5 milliseconds. This is the altered region of arbitrary transform, and then eventually there is some inactive or passive transform, because the cargo can pass through to the different region. So I think this is the first time that is shown that there is a correlation between the motion of one protein in the nuclear pore complex and the transmission of a cargo from one side to the other. And that motion occurs in the same time scale that we expect for active transport, and cross-correlate with the color, which is the cargo is colored. So I think more or less this is the summary. So we present an approach based on the pair correlation function and the particle tracking, in which we can see the motion of a particle and cross-correlate the motion of the particle from one side to another of the nuclear pore complex. And this protein has this characteristic to be either compact or large or long. And that is not new. There is a lot of other examples in nature in which you have polymer can be extended or contracted, And okay, so that seems to be one of them. Why we choose nucleoporin 153? Because it's a long protein, because it's anchored in the basket, can extend up to the other side and then come back. Of course, that does not explain that this is the motor that changes things, because there are many other things. So clearly it cannot be a process based on phase separation or phase, because that will require too much time. So the cross correlation as a hint, like a spring-like mechanism, so extended or not extended, extended or not extended. Okay. Now we did those experiments. He inhibited the NUP 153, and there is no more traffic, no more thing. But there is still a really large problem to solve, which is why the nuclear pore complex has 8 subunits. Are all 8 working independently, or are all 8 working all together to transfer things from one side to the other? And we don't know about that, because we mark only 1 out of the 8 nucleoporin, and might be, we should mark more of them and see cross correlation between the motion to answer that question. So clearly, we have made a step forward that we need this kind of pair correlation in order to see the motion of molecule. We can see the transfer of the cargo and many other things, but we don't know if that is really the mechanism or not. So I conclude right here, because I believe this is a really very interesting result about correlation function that can be have a peak instead of being just a relaxation like occurs in diffusion. So this is just to show that there are different kinds of correlation function, and then shows a possible mechanism for the transport to the nuclear pore complex. I finish here.